All right. Without further ado, hi everybody, and welcome to our next I Am Journal Club. I'm your host, Ben Geisler. I'm a hospitalist at Mass General Hospital in Boston, Massachusetts, and I'm also a researcher at LMU Munich in Germany. We are incredibly privileged today, as we could not have a better discussant. Dr. Isaacs is a POTS virologist at Penn, and among many other works, the author of the UpToDate crowd on the topic. I will introduce him in more detail when we get to the Q&A. Dr. Isaacs, as I explained to you, our audience here is a mix of clinicians, mostly hospitalists, MDs, DOs, and NPPAs, and other general interests, with a few internal medicine residents and also a few ID-trained docs sprinkled in. And we also have some epidemiologists, statisticians, data scientists, and public health folks. So welcome, everyone. I will, after some very brief background, present a case series on monkeypox patients from the UK. I will really try to fly through this so that we have as much time as possible for the Q&A. If you'd like to re-watch it, we will put the video on YouTube. And you can already start putting questions for Dr. Isaacs in the chat, and we will get to them when we start the Q&A. So back to the study type, it might be a little bit unusual to Journal Club a case series, but you guys might remember a case report, a case series from Munich, Germany, about the first recognized transmission of SARS-CoV-2 from an asymptomatic individual, or just to say that a case series may be helpful in highlighting very early trends and features of a disease or a syndrome. So in terms of background, even though monkeypox may have been discovered in Denmark in monkeys in the late 50s and later in humans in the 1970s in what is now Congo, it seems fair to say that monkeypox has been, until very recently, a neglected tropical disease. Past cases in developed countries have mostly occurred in returning travelers or through imported animals, except for a small human-to-human outbreak in the US in 2003. It's in the same genus orthopox virus as variola, the causative agent of the now eradicated smallpox, as well as cowpox, the OG smallpox vaccine. And so monkeypox is this brick-like virus particle of virion-containing double-stranded DNA. Under an electron microscope, it might be indistinguishable from other pox viruses. The externally enveloped virion features various proteins that might either facilitate entry to the host cell or interact with the immune system of the host. So the reservoir is mostly mentioned as being rodents, though as discussed on a recent episode of This Week in Virology podcast, this may not be definitely proven yet. Also, it would raise the question if pets could harbor the virus. In Western and Central Africa, monkeypox had been an endemic disease with two different clades. The Central African clade seems to have more effective IL-1 beta binding protein, which might be better at suppressing the human immune system response and might therefore have a higher mortality than the West African clade. Per the latest ProMed mail, there are now over 300 cases in over 20 countries recognized, which per WHO official is unusual. She said, we normally have only a handful of cases exported to non-endemic countries. We have unknowns about the origin of sudden outbreaks in non-endemic countries. And they also said that the current outbreak can be contained if we act now. 100 of these cases seem to have occurred in Great Britain and about 25 in Spain. There is speculation that some of these cases are secondary to index patients who attended LGBTQ events in Spain and Belgium. As is public knowledge, we have had a case of monkeypox at the Mass General. This was a case with likely transmission within the US. And there was a plan to vaccinate the clinicians caring directly for the patient with a specific monkeypox virus vaccine at juniors. And now we will discuss the clinical features and management by a recently published case series. The first author is Dr. Hugh Adler from Liverpool, United Kingdom, and the case series was published less than a week ago in the Lancet ID. Thank you to Dr. Nino Neumann for suggesting the paper for this occasion. The authors describe all 7 cases that occurred in the UK over a series of 37 months prior to October 2021. They used a PCR of various tissues and fluids and described the response to 2 different antivirals. These are the baseline characteristics. As you can see, 4 cases were acquired in Nigeria, in West Africa, which may explain the full recovery of all patients, and 3 in the UK, except for an infant. Everybody was between 30 and 40 years old, and therefore not vaccinated against smallpox. Aside from lymphadenopathy in 71% of cases, patients had between 30 and 150 concurrent lesions over skin and mucous membranes in the entire body. Complications included adjustment disorder and delayed healing of lesions, including deep tissue abscess in 1 patient. And DNA was isolated from various, virus DNA was isolated from various body fluids. Not mentioned is, but on other graphs is PCR from unroofed skin lesions. So you see a couple of sample lesions here. A and D show vesicles or pustules. The ones in F and G may be a little bit more subtle. And then in B and C, you see more macular appearing lesions on palms and soles. And then D and E here show subankle lesions, and the one in E here is maybe already stepped over. And then in H here, you see, in panel H, you see an ultrasound that was obtained while draining a deep abscess that complicated the course of 1 patient. The authors summarized the causes of their patients in these colorful graphs. You can see 1 patient here with an active skin lesion, and within 21 days, the presumed maximum incubation period is 21 days. The next patient has skin lesions already. And in these patients, you can see that various PCRs were obtained and from various fluids. Here again is mentioned blood and upper respiratory tract. On these graphs, you can see the PCR cycle threshold, or CT number, which you may be familiar with from COVID-19 over time. And the higher the CT number, the less virus. So black is the ulcerated lesion, and the other lines indicate other bodily fluids. And then the dashed line here is indicates when the rash was present. The gray area is when they were under the care of their special pathogens team. And the black arrows here indicate here and down here indicate when an antiviral was given. In patient 1, only 1 dose could be given, and in patient 2 doses, brincidofovir was the one given in these 2 patients. But in these 2 patients, only so few doses could be given because they had LFT elevations. You see here the ALT over time, and you can see the first dose to patient 1 in gray circles here given spikes shortly after the first dose, and then patient 2 is in in blue, in green rectangles receives 2 doses and then also has a spike in their ALT, in the serum ALT. There were also 3 patients that did not receive antivirals, and 1 patient who received another antiviral, tecovirimat, which did not have to be stopped for adverse events. Length of stay was between 10 and 35 days, and all patients had a full recovery. So some quick takeaways from the study: a direct contact, not necessarily during, but also before, after, or even without sexual intercourse seems to be necessary for most human-to-human transmissions. Droplet transmissions may be from possibly unrecognized mucous membrane lesions. Skin lesions may at first be subtle, but it's important to examine the entire skin, including palms and soles, and consider sending various tissues and fluids, including from the unroofed lesions, for PCR. The antiviral cidofovir may cause LFT elevations, which may prompt it to be stopped. Remember that patients may or may not develop a deep abscess and may have severe pain from this. Unclear what causes this. PPE will, of course, be prescribed by your infection control team, who may also contact trace cases, and obviously involved should be special pathogens teams, which is kind of the perfect segue for today's discussant, Dr. Stuart Isaacs. Dr. Isaacs attended Brandeis University and Yale University School of Medicine. He trained at Temple School of Medicine in Internal Medicine and Tufts School of Medicine in ID. At the National Institute of Allergy and Infectious Diseases, Dr. Isaacs did a postdoc in molecular virology. He runs a pox virus lab at the University of Pennsylvania, where he currently is an associate professor of medicine and has held various leadership positions, including associate dean for animal research. He attends at the Philadelphia VA, where it is currently his first day of service. And when we contacted him, Dr. Isaacs stated that it was certainly an interesting time to be a pox virologist. You can only imagine that he gets a lot of emails and requests. So bearing that in mind, as well, that it's his first day of service, it is absolutely wonderful that he could join us today. Dr. Isaacs, thank you so kindly.
Yeah, thanks for the invite. That was a great summary of the cases, and happy to be here.
So maybe I can get us started with the question. Please feel free to put questions in the chat. The most obvious question on everybody's mind might be what is causing the current outbreak? What is different about the virus or the circumstances that surround the transmissions?
Yeah, hey, thanks so much. Yeah, that is the $1 million question right now, and this is very unusual. I'd agree with what the WHO people are saying, that this what's happening now is not to be fully expected. And just to clarify on the cases you summarized that the UK published, because we all need more information about human monkeypox in the Western hemispheres, those cases are not really typical of what the cases we're now seeing here. So I'm not sure about the sexual transmit, that statement you said. So that was, clearly cases who got infected in an endemic area and then traveled and either came down with the illness or were getting sick on the way to the UK. And then the transmissions were, I think, within a household, and then someone in the healthcare system came down with it through potential handling of infected materials without proper PPE on. So those cases, if all the cases going on now were clearly imported cases from endemic areas, I think we'd be less nervous and less were raising alarms about what's going on. It's the current cases that, having so many countries and then with apparent infections in people who had not traveled, so showing some community spread going on. And so this is all quite unusual, and we need just a lot more epidemiology information to understand the interconnectedness and where these people might be acquiring monkeypox.
Yeah. And so aside from what was contained in that case study, what is your message to clinicians on the call? What should they bear in mind when they see a suspected case of monkeypox?
Yeah, well, and the great with the great education that's going on now to even have people put monkeypox in their differential when they're seeing someone coming with lesions, and now more so focusing on that a lot of these cases were presenting to STI, sexually transmitted infection clinics, where there were lesions in the genital area, and potentially that's the only place where lesions were genital, rectal areas. So, to even raise the potential could this be a monkeypox and not, syphilis or not herpes? And that's one thing in raising the alarm, to have people think about it, potentially test if there are no other known diagnosis to explain this. So what I really think, and not to make the same mistakes that were made during COVID, is to have the ability to test for this pretty easily and not require, like in the US, all samples are going to the CDC for confirmation testing. But I think it would be really useful to have more widespread ability to test to say, is this an orthopox virus, to at least begin to quarantine and tell patients to limit contact, to not have contact with others.
Mm-hmm. And there's one question, will we be able to stop the epidemic without vaccination of risk groups or larger groups of MSM men who have sex with men, or will we need vaccination of these groups?
Yeah, it's going to be too early. I think it's too early yet to say what the public health response is going to be to this, because if you could stop the transmissions by isolating or just telling people not to have close, intimate contact with other people, you can stop the transmission train. But if there's more and more community spread without knowing sources of where this is going, then I think there would be a more dramatic public health response to begin to do ring vaccination, not do widespread population-based vaccinations, but to try and vaccinate communities that are having this close contact. And once again, that's part of the epidemiology that we really need to define, is where these people are getting the infection.
Mm-hmm. And a related question, should healthcare workers be vaccinated? And again, if who? Yeah,
no, another good question. In preparedness, and this in the United States back in post 9/11, and then with the anthrax attacks that were bioterror attacks in the United States, where anthrax was sent through the mail, and there were a couple of deaths and infections from anthrax. There was a big response similar to what, maybe not as big as what we've seen to COVID, but a real thrust of resources for research and development of countermeasures. And at the time, they were most concerned about anthrax and smallpox. And that's really where some of these therapeutics that were mentioned, though the pipeline for those therapeutics were developed during that time in the early 2000s. And then in more recent years, those drugs got FDA approved here in the United States. And similarly, a safer smallpox vaccine was also important to have in the stockpile. And that's how Jynneos was ultimately chosen as the third generation smallpox vaccine. And this is an incredibly safe vaccine, has to be given as a prime and a boost. So the historical smallpox vaccine is a single vaccination, gave pretty prolonged immunity, certainly from serious smallpox and death. But Jynneos needs a prime and a boost, but is a very much safer vaccine. And I think really fortunate that we have that in our toolkit, because it's a much easier vaccine to suggest giving to people than the historical vaccine, which, we see what the reactions have been to getting COVID vaccinations, which are incredibly safe and very cool. The historical vaccine, I think, would be a tough sell for people. So it's really fortunate that we have Jynneos.
Yeah, I guess the adverse events are way more frequent with these original smallpox vaccines than with modern COVID-19 vaccines, for example. In terms of treatments, the two antivirals that were in that case series,
Brincidofovir, yeah.
Did you have a preference for either of them? One of them, Brincidofovir, seems to have been implicated in these LFT elevations that were in the slide deck.
Yeah, so I do have a preference of the tecovirimat, which is I saw that drug develop from its very infant days in the world of pox virology. You showed in your introductory slide some of these, the pox virus life cycle is very cool. It makes two types of infectious virions: this extracellular virus, which has an extra viral membrane on it, and a mature virus, which also has a membrane. So it has a single membrane virus and then a doubly membrane virus. And during pathogenesis, both forms of virus are really important. It's thought that the mature virus, which is the virus that can be dried down to a powder and used as the vaccine, is the virus that's probably transmitted. It's the virus that just builds up in a cell and you become like a balloon filled with marbles, and that's all mature virion sitting in that cell that either gets burst open or gets coughed out and desquamates and dries and is the form of transmission. Those pustules are just chock full of mature virus. The extracellular virus has this extra membrane. It buds through the mature virus, buds through the Golgi, and then gets exocytosed from the cell. And that virus turns out to be really important for spread within the host. That if to infect the next cell or distant cells, that EEV, that extracellular virus, EV, is really important. And tecovirimat shuts off the production of that extracellular virus. So you still make all this tons of virus in the body, but you're not allowing the virus to spread within the host, and it allows the host immune system to catch up and kind of take care of the infection. So tecovirimat is cool. The brincidofovir, which is a analog of cidofovir, was I forget the chemistry to make a orally bioavailable cidofovir, and brincidofovir was then developed for pox virus, as well as the company tried to get other indications for other viral infections. And so it inhibits the polymerase. And the dose needed is to show activity is fairly high, and I think could be part of the reason why you have the elevated LFTs.
Mm-hmm. Yeah. And then this question may be a little bit provocative, but do you think there is just better surveillance? Maybe there's cases that are now recognized that might have already been there in the past, or is this clearly higher numbers than we've seen in prior years?
Well, yeah. So, certainly what I think most people are now seeing, given the numbers out there, is that there isn't clear connectiveness between each of the cases occurring now, is that there may have been a introduction of this virus earlier than the initially reported cases, and that there's in some community groups, there has been some ongoing transmissions going on that were not detected until now. And that's kind of why we continue to see identifying cases, because there may be some other pockets of it. And so what's really interesting is that we have had over the years importations of monkeypox in infected patients without this community spread. And so what potentially has happened now is it's gotten into a certain group where the transmissions from close, intimate contact is happening more efficiently, and that just didn't happen before. You had mentioned the first outbreak of monkeypox outside of Africa in the West was in the United States, and that was related to the importation of some exotic animals from Africa for ostensibly for pets, that people wanted these exotic animals. And then in a distribution center, those animals came in close contact or direct contact with prairie dogs, which also in the United States, certain communities use as pets. And turns out prairie dogs were incredibly susceptible to monkeypox. And so prairie dogs were infected by the rodents that were imported from Africa, and then the prairie dogs were distributed into households and became sick and bit or scratched owners, and those owners came down with monkeypox. So, but in that case, almost all the cases were direct. You could show directly that they had interactions with the sick prairie dogs. There was a lot of veterinarians and veterinarian techs that also got infected from people bringing their sick animals to the vet. And there was really no clear evidence of any human-to-human transmission in that 2003 outbreak. And the outbreak cases were identified. It went a typical outbreak curve where cases were identified, they peaked and came down, and there were no further cases of monkeypox. But something very different is happening right now. That curve is still going up. It hasn't started to come down, and that's what's raising all the concerns.
Fascinating. We have another question about vaccination. Should we support endemic countries in vaccination programs against monkeypox to avoid new epidemics?
Yeah, so much of this was all predictable in the endemic countries, that during the smallpox eradication program and during widespread smallpox vaccination, that protected against monkeypox. And a decision was made after the eradication of smallpox was not to continue vaccinating with smallpox vaccine in Africa. And part of that was due to the historical smallpox vaccine and the adverse events and things that were seen with it that they were worried, I think, of doing more harm than good. But once you stop vaccinating and the population no longer has any immunity to the orthopox viruses, it was predicted that there would be a rise in cases of monkeypox in Central Africa and Western Africa. And epidemiological studies have clearly shown that there's much more human monkeypox cases due to a combination of both, environmental factors where people are, getting closer, living conditions are getting closer to jungles where they may come in contact with animals, and then the travel of people from jungled areas into the more populated cities where there's more and more human cases. And so the CDC is actually doing a study right now with Jynneos in Africa to test. It's not a randomized controlled trial. It's more of an observational study of people who are being vaccinated and comparing it to historical controls. But that is a question whether it will make sense to vaccinate in the endemic countries to prevent these animal to human transmissions that, over time, the more and more introductions into humans, there could be the, concern of mutations happening that would make it a virus that can more easily spread person to person than the current virus.
Yeah, and I'm sure the maker of Juniors is looking into ramping up production. Probably in the past, they've only worked to supply the national strategic stockpiles, but now there's, at Mass General, they thought about vaccinating the healthcare workers in direct contact with the patient. And then there might be countries who are interested in purchasing more vaccine. Do you happen to know how expensive it is at the moment?
I do not. The U.S. and I couldn't even tell you what the U.S. government paid for its 300 million dose stockpile here in the U.S. I do not know that. But my understanding at the Mass General, at least, and since you guys are connected with it, you may know more than I. But I thought they were risk stratifying people, at least who were in contact with the more recent patient who will spend time in the hospital for a number of days before a monkeypox diagnosis was made. And so they risk stratified people and then offered vaccine as post-exposure prophylaxis. I really, and I get the question a lot about, well, should we be vaccinating doctors, ER dermatologists, sexually transmitted infection workers who man sexually transmitted infection clinics? Should we vaccinate those prophylactically? And, again, I'm not sure we're there yet. Especially in this time of COVID, I know when I'm having face-to-face contact with patients that I am seeing, even non-COVID infected patients, the PPE we're wearing now routinely, which includes eye protection, a face mask, not always a gown, but certainly gloves, where, in the US, as clinicians, we're not wearing masks. We were not always wearing eye protection. So I think the risk of a clinician or a healthcare worker who's interacting with the patient in a normal encounter where the patient's usually masked, the provider is certainly masked, that it's really going to be unlikely to have a exposure in that setting. I do worry about some of the ancillary staff, the people who clean the room, change the bed sheets and all. Someone with active monkeypox or any of these orthopox viruses, the virus is just desquamating off their skin, off of those lesions into the bed sheets and their clothing and things like that. And I think those were the more high risk exposures that occurred at the Mass General with that index case that recently was there.
Mhm. You mentioned these prairie dogs who were infected during the 2003 outbreak in the US. How do you judge the risk of this becoming endemic in Europe, Northern America, and other parts of the world by infection through local animals?
That great question. What we're saying is a reverse zoonosis where humans are the vector and they infect animals and it brings hope. And that is a concern. It was certainly a concern in 2003, especially with the prairie dogs and with the news going out about how these infected prairie dogs, public health was very concerned that people were going to just release the animals into their environment and get them out of the house. And there was definitely concern that some other species would then become a good host for monkeypox outside of Africa. That was carefully watched, looked for no additional human cases, but I think animals were also captured and looked for to see if there was any evidence of transmissions out in nature, and none was found. So that's reassuring. This number of patients currently out there right now, there is a concern about transmissions to a species that would then be able to continue to propagate it. That was actually seen in back in the day of smallpox eradication with the smallpox vaccines, this live growing vaccine that was a scarification on the arm that was exposed, and you could spread it to other people who you were having close contact. But there are some animal reservoirs of the old smallpox vaccines in India and South America where these animals have pox lesions now 30 years later. And if you sequence those viruses, they're most related to the vaccines that were used back during smallpox eradication. And that was all felt to be from human contact with their domesticated animals, and then transmissions that then persist to this day. So that's certainly a concern, and hopefully not an outcome of the current outbreak.
Mm-hmm. Speaking of sequencing, there probably haven't been that many genome sequenced in the current outbreak, but there seem to be some early indications. I think there were some genomes posted on a website called Next Strain, and a discussion on virological.org, that there have been quite a number of mutations in the virus implicated in the current outbreak. Over 40, while this is usually a pretty slowly mutating virus. Could that explain that this, could that mean that this virus is possibly more transmissible?
Yeah, so I have not seen that. The information I'd seen was just that at least some of the initial sequences were that these were from West Africa, from Nigeria, similar to strains back in 2018 and 2019, and so that was good. Western Africa, a less virulent strain than what's being seen out of Central Africa. But, so all viruses have some, this is a large DNA virus that has a high fidelity polymerase that doesn't like to make errors. But errors, these viruses replicate rapidly, and errors are made. And I think there's always going to be some, point mutations. I'm not sure what mutations have been described, but the thought right now, given that it seems like the infections are from very close almost skin to skin contact that I don't know yet if we could say, oh, this is spreading through more casual contact, and that would be more concerning.
Mhm. And then there's another question about treatment. The question reads, who really needs tetraviromet? In that case series, I know that they only had very small numbers, but the 2 out of 7 patients who weren't treated with an antiviral also did fine. And this is the Western African strain.
So 2 important points is people with monkeypox don't need to be hospitalized most of the time. So what's, is part of this hospitalization just to ensure isolation. And then most of the time, most patients don't need treatment. This is a it will heal on its own. But from what I've heard from the current outbreak, is that some of these patients have very painful lesions in various parts of their body. And by treating, the thought is to begin to allow healing to occur more rapidly. So that may be an indication for treatment, would be someone who has extremely painful lesions that you want to start treating. The other is if it was gotten into an immunocompromised person, very young patients might have a poorer outcome and may be someone you may want to treat earlier rather than just allowing it to take its course. But you're right, most of these are not being treated, do not need treatment. And I think part of it is us as doctors, we want to, an infection is there, we want to do something and treat the infection, and you don't always have to treat.
Yeah, that makes a lot of sense. This deep abscess that was in that case series, that probably also surprised you, or have you heard of anything similar? Is this something we should look for if somebody really has very painful lesions?
Yeah, I mean, I've heard of cases of proctitis where the lesions are there, very severe lesions around the mouth has been another case I've heard about. But yeah, so, it's interesting. Back in the day during smallpox, a lot of these lesions became super infected, and how much of bacterial infections also led to the morbidity and mortality of smallpox during the centuries that it plagued humankind is a good question, because certainly in some animal models with certain pox viruses, not specifically orthopox viruses, but sometimes the bacterial superinfection that these animals get actually lead to death, not so much from the virus, but from the bacterial infection. So a deep infection like this, I'm not quite sure the pathogenesis, whether it began as a skin lesion that then went down instead of up as to cause the bacterial infection in an abscess. But then also, as you saw, you have viremias occurring with these pox viruses, which is really the cause of the widespread rash when it does happen. It happens from a viremic phase of the virus. You could imagine that that viremia, if it's also carried in cells, that a abscess or a fluid-filled area might be an area where this gets inoculated, and find it there too.
Is blood a more sensitive bodily fluid to your PCR than vesicle?
No, probably not. No, the vesicles is definitely, you're going to find virus there. The blood is looking at some of those graphs, not everyone was it's sometimes hard in experimental animals. It's sometimes hard to find the persistent viremia. So we have animal models where we intravenously give monkeypox to non-human primates to test therapeutics and/or vaccines. And so certainly giving it that way, you get a viremia, and then over time that viremia comes down. So the lesion, if the lesion's available, that's where the money is for a diagnosis.
And then our final question is, do you think there could be asymptomatic transmissions?
Yeah, that's another great question. And I don't think we know enough yet. I mean, one of the reasons smallpox was a disease that we were eradicate was there was no asymptomatic spreading of disease, that anyone who got smallpox who could spread it develops lesions, and then you did the ring vaccination and you were able to squelch it. So in Africa, if during seroepidemiological studies to kind of see how there's more antibodies against pox viruses in Africa than there is people saying, oh, I had monkeypox. So there may be a mild disease and/or potentially asymptomatic disease. So that could exist. Now, whether that is able to spread is a question we don't know, and it's thought not, but could that also be the explanation for this current outbreaks? Because during close intimate contact, if someone has obvious lesions, I would think people would hesitate to have any that type of contact. So that's a good question, and I think we don't know the answer yet to that for the current outbreak.
Well, thank you to the audience for the great questions, and of course, thank you so much, Dr. Isaacs, for spending some time with us. That was incredibly illuminating. Thank you again.
Appreciate the invite. Love talking to my colleagues in Boston, around the world, and we'll see where this goes. This is quite fascinating, and as I said as Ben said, it's an interesting time to be a pox virologist right now. That's for sure.
For sure.
So, thanks for the invite.
Thank you. Bye now.
Bye all.